Hello, everyone. Thank you so much for joining us, and welcome to yet another edition of the GSU Malaria Journal Club, which is a monthly series. I see people are trickling in. Welcome, everyone. And I do know that we do have audiences from across the world, Africa, the US, Europe, and everywhere else as well. So thank you so much for joining us in your different time zones. Today's webinar is a parasite-focused one, referencing Dr. Lyimo's recent paper titled Genetic Polymorphism and Evidence of Signatures of Selection in the Plasmodium falciparum circumsporozoite protein gene in Tanzanian region with different malaria endemicity. My name is Pablo Abel-Chauke, for those of you that do not know me, and I'm a training coordinator at Malaria Gen, GSU, working with a team of awesome people to organize these monthly events. Just a few ground rules. The Journal Club is being recorded and will be available later on our YouTube channel, on the Malaria Gen YouTube channel. So please do check it out and share with your colleagues and friends, and do rewatch for some lessons learned from Dr. Lyimo. Please stay on mute so as to not disturb our speaker today. We will have a Q&A session as well as discussion at the end, where you can ask questions either by raising your hand, or we'll unmute you so that you can speak, or you can type your question or comments in the chat Q&A box as well, and we will read them for Dr. Beatus Lyimo. Without further ado, I would like to introduce our speaker for today, Dr. Beatus Modest Lyimo, all the way in Tanzania, who kindly agreed to give this webinar when I asked him a while back. So thank you so much, Doctor. Just a bit of a brag. Dr. Beatus is a graduate of our Anopheles Genomic Surveillance Data Analysis training course, which just completed last month in June. And just to report, he was very dedicated. So he did receive a certificate of completion. So well done to you, Doctor. The title of the talk is, as you can see on the screen, is similar to the paper as well. Just to let you know about who Dr. Lyimo is, a bit of a short bio about the work that he does and his experiences. Dr. Beatus Lyimo holds a PhD in life sciences with a background in molecular biology. He currently works as a lecturer at the Nelson Mandela African Institution of Science and Technology, NMIST for short, in Arusha, Tanzania, where he teaches genomics and bioinformatics as well. From 2020 to 2023, Dr. Beatus worked as a postdoctoral fellow in bioinformatics at the National Institute for Medical Research on a project titled Molecular Surveillance of Malaria in Tanzania, where he worked to understand the population structure and drug resistance of Plasmodium falciparum parasite that is circulating in Tanzania. Before his current post, which he currently holds, he had been a senior scientist at the Nelson Mandela African Institution of Science and Technology in Tanzania since 2011. His research interest is to contribute to human capacity as well as the health system through next generation sequencing, or NGS for short, as well as bioinformatics approaches. Dr. Beatus envisions a future for Africa where NGS-based and bioinformatics strategies are commonplace for health research systems. Beatus, thank you so much for joining us. You may begin your presentation.
Thank you, Pablo, and your colleagues for inviting me to present in this Today Journal Club. Thank you, everybody, for taking time to join our Today Journal Club. And good morning, good afternoon, and good evening, wherever you are. I'm so delighted to present this work on the genetic polymorphism and the evidence of signature of selection in Pfcsp circumsporozoite protein gene in regions with different malaria endemicity in Tanzania. As I've been introduced, I'm Beatus Lyimo, and I'm from Nelson Mandela, which is based in Arusha. And I'm going to take you through this presentation on the paper which we just published a few months ago. And I'm going to present on behalf of these good people which are listed as co-authors here. And before I go down to the presentation, which are going to highlight some findings on the genetic polymorphism of CSP in Tanzania, which we just recently published in support of everybody which are listed in the as co-authors. So I thank the co-authors and I thank them for allowing me to represent them in this today journal club. So I'm going to, my presentation has a few highlights. Going to have introduction, going to have methods, research and discussion, future direction, and acknowledgement. In an introduction, as most of you are coming from the malaria field, you may be aware of this, some of the picture and the figure which I'm presenting here. But it's important for us to keep ourselves reminding what is going on in the malaria burden, as it's our all aim and the focus toward the elimination of malaria. So whenever we get time to discuss malaria, it's very important, again, to remind how far our struggle or our fight against malaria have reached so far. So from the WHO report 2023, it still shows that malaria is still a very high burden. And when we compare the malaria death and case from 2019 and 2022, you can see that there are both cases and deaths are keeping rising. But when we compare the cases per 1,000 population and 100,000 population, still you can see the problem is there. And you can see under 5, still the incidence and the deaths are still very high, which it's important to know so that we can increase our effort. Further down, we can see that that malaria is still a big problem globally, including Tanzania. And half of the world population is at risk. And by last year, from last year, we can see that 259 million cases and 608 deaths were reported in 2022. And surprising that 94% of all cases and 94 and 95% of both cases and deaths were reported in sub-Saharan Africa. Another thing to note is that at least 93% of Tanzania population live in an area which they can contract malaria, which is very high. Almost everybody is at risk of getting malaria. So what hinders from achieving the elimination? There are several factors which we need to consider and see them. And as we are setting our strategies, we need also to remember and include them during the elimination strategy. One of which is the issue of anti-malaria, which is keeping rising every time and then. And we are still reporting the new case with ATC, which is coming closer to East Africa. So it's still a very big problem. We have the issue with insecticide resistance. We have issues HRP2 region deletion. We have a new case of invasive vector parasite, which has been reported in East Africa. Also, we have the issue of limited financing. We have the issue again of climate change, where there are some areas where the malaria was 0, but now we are starting to see the number of cases are rising with the rise of temperature and a new breeding site for the mosquito, which provide a favorable condition for the mosquito to breed and replicate the parasite. So we also have an issue of limited diagnostic capacity in our health settings. But there are issues of genetic variation in parasite and vector is another challenge to consider. And there are also issue of social cultural inheritance, which especially under use of insecticide treated in age and related issue. And also we have the issue of lack of effective vaccine. And our today topic will be much based on the vaccine, especially the 2 vaccines which have been approved by WHO. So we all know that WHO recommends 2 vaccines for the problematic use in children from moderate to high malaria transmission area. And we know that this RTS, which have been approved in 2021, and the R21 matrix, which was approved in 2023. So the 2 vaccines have the closer structure. That's why I wish to study both of them. That the RTS and the R21 are both bound into hepatitis B protein, but with RTS, it has a very like 1:4 ratio of CSP protein and hepatitis B protein. While for the R21, it's covered like bond like 100% with the hepatitis B protein. And this it's justified that the R21 may have very high efficacy and elicit high antibody response compared to R21, compared to RTS, as we see that it contains a high proportion of CSP protein. And we further report that when we compare the 2 vaccines, that the RTS has the 2 portion of hepatitis B, while the R26 has only 1 part. So in a natural condition, that it's important to know that Pfcsp contains 2 regions. The one which has the amino acid repeat, which is a highly polymorphic, and the C-terminal region. And this Pfcsp is a predominant surface protein of sporozoite, and it plays a crucial role in the invasion of hepatitis C or the liver cells. So this makes this gene a key target for malaria vaccine. And from previous studies, it will be revealed that there is high polymorphism in this region, especially within the C-terminal of the Pfcsp gene in the natural parasitic population. Now, the genetic polymorphism of this gene, it raises concerns about the strain-specific immunoresponses and the long-term efficacy of this vaccine. And from the studies which have been reported previously, they show that vaccination improves protection with high efficacy for 3D7 matched compared to 3D7 mismatched alleles. So that, due to that, it's make us come up with this study. And the aim of this study was to explore the extent of genetic diversity and the natural selection of malaria vaccine antigen and Pfcsp in natural parasitic population. In order to generate, this will help us to generate baseline data for Pfcsp diversity before the introduction of malaria vaccine in country. And this data which will be generated will be used to monitor the performance of malaria vaccine post introduction of the vaccine. And we use the data which we are collected from a cross-sectional study. And this study took place between May 2013 and January 2015 from 5 regions of mainland Tanzania. As you see from this map, we have Morogoro and Tanga, which we are considered as moderate transmission areas, and Kagera, Ujiji, and down here the Nachingwea, which are considered as the high transmission regions. And the DNA was extracted and shipped to Sanga, and the whole genome were generated and deposited into the malaria gene database. So from this study, which we did, we downloaded the whole genome sequence data from a malaria database, F7, and we had a good sequence of about 590 sequences. And we did all the cleaning to ensure that we have a high quality sequence. And we chopped all the gene, which is the Pfcsp gene, which is located under the chromosome 3 using the BSF2 software. So from that data, or the clean and the high quality data, we analyze the FST and the PCA so that we can measure the population differentiation and the structure. And we also use the Tajmak-D from the VCF tool to measure the evidence of signature of selection. And we also measure the gene diversity and the haplotype diversity using the DNSP software. And we draw the haplotype networking using the network software under the median joint algorithm. So after that, let me report the result which we got from our findings. From the measure of the monoclonality, which we use the FWS, we found that in Nachingwea and Lindri, which is the southern, Nachingwea Lindri, which is the southern part, had like 69.2 polyclonal, while Morogoro had 68.75. Muleba had 63%, 24%, while Ujiji had 63.46. Only Muheza in Tanga, which is a moderate transmission, had a high proportion of monoclonal infection, which is like, it was 43.1%. But when you use the chi-square to measure if this variation we are seeing here, it has any significant difference, we found that it was not statistical significant. Yes, there is variation which we observed, but the variation was not significant difference. We also measure the genetic differentiation and structure. As I mentioned, we use the FST to measure the genetic differentiation across the 5 regions. And the result which we got, it was 0.068, which is less than 0.05, indicating that there is no that much variation between the 5 regions. And this was further quantified by the PCA, which show that from the PCA which we are seeing here, there are no any population structure. So most of the samples from all 5 regions, we are clustered together with a few samples which we are like outliers, but most of them were clustered together. This indicating that this species, all these individuals have there are a continuous gene flow among the population, and there is high genetic exchange despite the genetic separation. From the map which I previously mentioned, you can see that maybe Muleba and Nachingwea are very far apart. And maybe we can expect, due to geographical any other factor, we can see some clusters or different, you can see a structure of Plasmodium in Tanzania. But from the result which I'm displaying here, or from the result which we got, we didn't see any structure, with the exception of few outliers. And we went down to see the structure of the C-terminal. And in this structure, we most consider interested to see the TH2R and TH3R. And we see that a lot of diversity, a lot of activity which are going there. And which suggests that the genetic variation were mainly concentrated in these regions. And this can be explained due to the biological part of it, how it contributes to the malaria infection. So there are a lot of variation which are taking place around this region. So we also try to see what are the other findings from the other regions across Africa. And we found that the pattern which we are seeing here, that we also reported in the other countries like Malawi, Gambia, Nigeria, Ghana, Mali, and Guinea, which is also when you find it globally, you can also find this variation. So it means there are a lot of activities which are going along this region toward the survival and the infection as concerned. We further measure the other signature of selection, like T and from this results, most of them show exhibit or shows like a slight positive, which show like a balancing selection in this most of the region, with the exception of Morogoro. And the haplotype diversity, which it was very not that much, it didn't show very much differences, but the overall of which was 0.937, which is closer to what is also reported in other countries in Africa. So it means there are similar to a nucleotide diversity and other parameter. They show that the neurodiversity was not that much different compared to other neurodiversity values which have been reported in other countries in Africa and globally. We also draw the networking to see how the haplotype in Tanzania when compared to the 3D7 sequence. As we all know, as we reported back there, that this haplotype or the vaccine was constructed based on the 3D7. And from the previous studies which have been shown that for the vaccine to work to produce a high immune response, at least should be a match with the sequence with the vaccine So the vaccine and the population should at least match to increase the immune response. So from what we got, we found that there's a very low matching of 0.2 and 0.02% matching compared between the circulating parasite and the sequence which is included into the vaccine. So it has been reported in the other country also that there is very high differences between the vaccine sequence or the 3D7 sequence and that which is reported into the natural population. And from we are discussing and see, is it possible maybe to come up with the vaccine which is regional specific, maybe East Africa specific or West Africa specific, targeting, or we have the vaccine which is targeting the circulating haplotype, so that hoping that this will be closer between the circulating haplotype sequence and which is the vaccine. Hence, we're going to increase the immune response, we're going to increase more protection compared to what is there. So this is what we see, but it's very also important to keep monitoring and see how the vaccine can be improved so that we can increase the vaccine efficacy. So in conclusion, we can report that no clear differentiation in the pattern of PSP gene chromosomes despite the study site being widely separated geographical. It means that we didn't see the population structure. And the C terminal of the Pfcsp gene display high diversity in the study area. And the haplotype diversity in the C terminal of the gene in Tanzania isolate was similar to those which were reported in other countries, in African countries. As I mentioned that we found notable differences differences between the haplotypes, which is designed for the RTS and the R21 vaccine, and those found into the natural population. And this may suggest that limited efficacy of the vaccine. So what is our recommendation and future direction? From the study or from the team, the assessment of the same gene using the most recent, which is 2023 data from 13 regions in mainland Tanzania. As what we reported only 5 regions. But now we're going to include more regions. It's a 13 region with more recent data and see how it's going to be the trend, be the same, with the variation. So this is going to help us to get more information. And more study in other regions to generate a baseline data in the entire country. As I mentioned, we're going to have 13, which we already have the data, but we wish also to expand to other regions in Tanzania, so that we can have a clear background baseline data toward the diversity of the gene in Tanzania. That's what we recommend for those other countries which are deploying or they will not yet start to use the vaccine, to have a similar kind of the study, so that they can have background information. Then this background information is going to help do the monitoring of the vaccine post introduction, and see that if there is any variation and if the variation what to be done. And also we recommend the study monitoring of Pfcsp gene polymorphism and performance of the vaccine in different parts of Tanzania and the other countries. Thank you very much, and that is it. And I would like to acknowledge all these people, including the government of Tanzania, National Institute of Medical Research, Nelson Mandela, the Brown University, other colleagues who've been working together. Don't forget the MSMT project, which fund this project and support my postdoc, but with a very good also support from Gates Foundation and Malaria Gen. Thank you very much for first allow us to use the data, but also to give me the chance to present what you found from the study to the other colleagues, and you can have time to discuss, to get comments. These comments, some of them I can respond, some I can take as comment for the future improvement of this work. Thank you very much.
Thank you so much, Dr. Beatus Lyimo. This was a brilliant presentation and really accessible and well explained as well. So well done and thank you. I'm reading the comments. Everyone is agreeing, saying great from Keen, good presentation from Mohammed. Nice work and excellent presentation. Thank you so much from Braxton and other people. Fantastic presentation from Desmond Domu. I'm not going to read all of the comments, but people are agreeing and saying this was a really great presentation. We are now at the time for question and answer as well as discussion. We have a bit of time, about 30 minutes to go. Just to let you know, again, as I said in the beginning, you are able to raise your hand and we can unmute your mic if you would like to speak or ask a question, or you can type your question in the Q&A box or the chat box as well, and we can read it for Dr. Beatus Lyimo. There's a question that I'm seeing now from Dr. Christina Ariani, who says, thank you very much for the excellent presentation. If you could fast forward to 5 years, what do you think the impact of the vaccines will be on the circulating parasites? Do you think there will be resistance? That's from Dr. Christina. Dr. Beatus?
Thank you very much, Dr. Christina, for the good question. Yes, for the next 5 years, we don't know what it's really going to be, but from what we see that there is very homogeneous gene which is circulating around the community, the local parasite, as we didn't see any population structure. It means that if there's any resistant gene which have been introduced into the population, it's going to be easier for it to circulate quickly around the population. And if it's really resistant and it can really, maybe it can affect the nature of the vaccine. But if we can have, maybe we can see structures, or we can see the closeness between the natural population, the circulating sequence, and that which made the vaccine. Yes, we can say that. Yes, maybe after 5 years the vaccine can be still working, but it's something which we need to make follow up. That's why we did the baseline study, so that we can have what is there before we start the vaccine to introduce, and we can make a closer follow-up, maybe after 1 year, after 2 years, after 3 years, what is changing, and you can measure it against the vaccine response. So yes, something can happen, but it is something which we need to monitor, to do monitoring, so that we can come up with a clear conclusion if there is resistant or something like that. Thank you.
Thanks, Dr. Beatus, for that answer. As I said, you can raise your hand if you'd like to speak. Otherwise, do type your question in the chat or Q&A box, but you can raise your hand and we will allow you to speak. The next question, Dr. Beatus, comes from Dr. Deborah Sumari, who's saying,
yes.
Thank you, Beatus, for a wonderful presentation. But you have said the vaccines may have limited efficacy in the Tanzanian population. So what do you suggest on the efficacy issues, since the vaccines have recently been registered for use, and basing in mind that the R21 has recently been registered and have not been widely used? That's from Dr. Deborah Sumari.
Thank you, Dr. Deborah, for your question. Yes, I agree that both RTS S and R21 have both registered, and now there are more than 28 countries which have ready to use. Maybe recent we have Central Africa, we have Malawi, and many other countries which are already show that they are interested to introduce this into their as part of vaccine to both children and which are in that endemic area. So based on your question, that some studies have indicated that yes, or from the preclinical trials result, we said there is some high immune response, especially for those which are matched to the sequence of 3D7, or those which constructed the vaccine. But with low efficacy, those which are being matched. And for the Tanzania circulation, found that they are very low matching, or there is high mismatching. Yes, it can be introduced, but the important thing is to do the monitoring. As we continue monitoring, we can see that if the efficacy can continue to be high, or we need to maybe to come up with some data and suggest maybe we can come up with a regional specific, maybe vaccine which is specific for East Africa or for Tanzania, so that to ensure that all of them have used the vaccine and they are getting very high protection, rather than use the same vaccine globally. So if using the same vaccine globally, yes, there are some losing and gaining. Yes, but to conclude, Dr. Deborah, yes, the vaccine can be introduced, but with closer monitoring to ensure that the performance is higher, and how the population is going to respond to the introduction of the vaccine. Thank you.
Thank you so much, Dr. Beatus Lyimo. There's a question with a raised hand from Professor Deas Ishongoma. Professor Deas, please go ahead and ask your question. You can unmute.
Thank you so much, Barbara. It was great for Beatus the chance to present to different colleagues and also get feedback. I just wanted to highlight some few issues, not a question, just a comment. So we need to first understand that the ones we have are not perfect. I just checked. The efficacy of R21 is higher at 77%, and that of RTS S is actually lower, below 55%. So that we don't have a perfect vaccine. And that's partly attributed to what Beatus said, that the vaccines were made based on different alleles, and these are not the same as what we see in natural populations. So to say we need to understand the baseline levels of the different alleles in the population and how they match with the vaccine even before we introduce them, because we have an opportunity and the tools to be able to generate the data and understand, and can now start actually to make predictions. If we, and this is what I wanted to plead to Christina and the Sangha team, if you can start to make predictions based on the alleles, we could now go to the second stage of monitoring the vaccines once they have been introduced to see whether they will match our expectations. So I also wanted to thank the Sangha team for the software they generated. And I wanted to request now, going forward, we map all the countries where we have the data and show how the vaccines will actually perform. And with the modeling, we can generate brilliant data that will help National Malaria Control Program and the global community to manage expectations of the vaccines, because there have been euphoria that vaccines will perform magically. So I just wanted to highlight that, and thank you guys for giving the opportunity to Beatus to present and share our findings. Thank you.
Thank you so much, Professor, for that. I don't know if Beatus has a comment on Professor's comment before I ask the next question.
No, thank you very much, Professor Shengoma. He's my boss, and he was worked together since the beginning of this work. So we went through all together up to here we are. And the recommendation, I really support them, that we need to come up with the prediction model. Now we have the different tools. We have artificial intelligence tool that we can can all use them to predict what will come next with the vaccine, with the efficacy, etc. So I really support the comment, and I really support the suggestion from him that the Sangha team can help us to come up with those prediction models based on the computation, bioinformatics, and the artificial intelligence from those data coming around the different endemic regions, so that tool can give us a clear picture, maybe in 2 years, in 3 years, in 5 years, what will be happening with the vaccine. So that is a very good comment, which I think everyone of us, we need to take it into a consideration for the future improvement and the performance of the vaccine. Thank you.
Thanks, Beatus. You are lucky to work with Professor Dias. He's an amazing human and amazing scientist as well. The next question comes from Flavia Bawa, who says, thank you for your presentation. There has been recent studies on the use of multiple epitopes for vaccine design. Is it possible to use this technology to make a single vaccine with all these variations using this technology? That's from Flavia Bawa.
Yes, thank you very much for that question, Flavio. Yes, I think also when we are thinking for the future vaccine, it's also important to consider all those different variations. And not only that, but you also can consider different candidates. Maybe we have several candidates which are in pipeline. So if we put them together, maybe 3 or 4 candidates in the same one to produce one vaccine, that's going to be very powerful, and maybe the efficacy and everything can be higher, and maybe it can help, or it can stay, or it can produce the protection for longer time compared to a single candidate. So yes, I agree. We can think of that. We can think of modeling. Now we have this, we call the in silico work, where we can use several candidates, and you can use a computational model to predict if AMA, MSP group, and the Pfcsp, when we mix them together and come up with a vaccine, how going to perform. So there are all those tools which are available, and they can help us to come up with the ideal vaccine. So yes, I support, the tools are there, and we can use them to come up with a very good vaccine. Thank you.
Thanks, Beatus. The next question comes from Honorine Kabore, who's saying, nice talk. Please, do you analyze the PCA using only the PF Pfcsp gene or whole genome data of the Plasmodium? That's from Honorine.
Thank you. As I mentioned during the methods part, from the whole genome, we narrow down to chromosome 3 because the gene is available in chromosome 3. Then from chromosome 3, we pick the gene. So we did the PCR analysis based on the gene. Got the whole genome, we're going to get a lot of information from the whole genome. We went to chromosome 3. Then from chromosome 3, we chop a specific region where the gene is located. Thank you.
Okay, thank you, Dr. Beatus. This next question might be related to the one that Dr. Christina asked. So yeah, I don't know if, but I'm gonna ask it. It's from Desmond Domu, who says, is there a possibility of drug resistance when the same vaccine is used in other jurisdictions without any proper analysis to know the genetic diversities? It was very informative and nice presentation. That's from Desmond Domu.
Okay, I did get the question clear. That can you come again with this question?
Sorry, is there a possibility of drug resistance when the same vaccine is used in other jurisdictions or regions without any proper analysis to know the genetic diversities? Yeah,
here we are talking of, maybe the question is talking about drug resistant, and here we talking about the vaccine. The issue of drug resistance, the Plasmodium can develop the resistance based on the what current vaccine, but some other resistance can rise de novo without any historic toward it. But with vaccine, with this vaccine, it's important, yes, you can use, but it's important to introduce or to have the baseline information. That will help the country or the one which are using the vaccine to do the monitoring, because it is very important to do the monitoring to see how they respond. Because as we know, the parasites do undergo several changes, and especially when they come across the new, maybe the vaccine, so they need to survive by themselves. So they may undergo some changes. Yes, so it is important to have the baseline information that is going to help during the monitoring of the vaccine performance in the international population. Thank you.
Thank you so much, Dr. Lyimo. We have more questions coming in, and I hope we have enough time to cover them. We have about 17 minutes left. So as I said before, you can raise your hand and we will let you unmute and speak. But for now, I'll keep on reading the questions that are in the chat and Q&A. The next question comes from Michael Maina. Good presentation. The question is, can we measure the immunogenicity of the circulating haplotypes as well as the 3D7 haplotype? Can we measure the immunogenicity of the circulating haplotypes as well as the 3D7 haplotype?
Yes, I think it's possible. There are several studies, one in Malawi and somewhere where they measure the immune response. And that's why because there are several serological tests can be applied to measure the immune response. That's why they managed to say that when you have maybe the vaccine with the matching with the 3D7, how the immune response was, and when it's mismatched, how it is. So that yes, it is possible to measure the immunogenicity, and there are several serological tools which can be applied to measure the level of efficacy, both the matching and mismatching. Thank you.
Thanks, Doc. The next question is from Dr. Richard Pearson, who says, thank you for the great talk, Beatus. What are the plans for vaccine rollout in Tanzania? Has this already started?
Yes, maybe I can say, and maybe also can, if you're going to allow Professor Shongoma to add on it. But what I know is that still the government is in the plan of introduce this vaccine in country. And when it's going to be introduced exactly, I can't tell. Maybe if Professor Shongoma can assist on that, but you know the country is in the plan of introducing the vaccine.
Thanks, Beatus. If I can add, it's true, Tanzania has not adopted and approved the use of the two vaccines, but we do have a national vaccine team that is advising the ministry on the best ways and how to go around introducing the vaccine. As far as I'm aware of, that the committee has presented a proposal to the ministry, and there have been some critical issues from the ministry which need to be addressed, like sustainability, funding, and also targeting. Recently, we confirmed that the missing partial resistance in northwestern Tanzania, and we have 2 papers coming in the Lancet Microbe and the Lancet Infectious Diseases next week. So please check out. And we are recommending actually deploying whatever we have, including the vaccines for all age groups. So the landscape is likely to change in the next couple of months, given the importance of fighting artemisinin partial resistance in Tanzania. Over to you guys.
Thank you.
Thank you, Professor Dias. The next question is from Anonymous, one of our attendees. I would like to know how long it takes for mosquito to develop resistance to the newly developed vaccine. Meanwhile, the treatment regimen being used is non-resistant drugs.
Maybe if I understand the question clear, that the mosquito, maybe to develop I can't tell you exactly how long it can take, but something needs to be established. Yes, I need that to say that I'm not very much sure, but because there are some variations which need to take place, so it's something needed to be established. So I don't like to say much about that, but something which we can also maybe Professor Ishongoma raise the question. Maybe you can give him the chance to respond. It's help.
I see Des has his hand up.
Pablo,
Des?
do you allow me to respond to that question? To contribute? Hello?
Yes, please go ahead, Professor.
Yeah, so I think for the listeners and colleagues, we need to be very clear what we are talking about. The vaccine targets the gene in the sporozoites, and these are infective stages of the parasites which invade the liver cells and start the process of developing in humans. So this has nothing to do with mosquitoes. And if resistance are to develop in mosquitoes, we should talk about insecticide resistance. But if we are talking about resistance to the vaccines, meaning that the vaccines can be there, but still not work, that should happen in humans. And it can happen, as Beatus said, for instance, if you are using a vaccine that contains an allele that was not in the original vaccine, means the vaccine will not have any function because it's you are using something against the other thing, which is completely different. It's like trying to open a door with a different key. But in this question, we're not talking about mosquitoes. We are talking about vaccines in humans. So we need to get that clarity.
Okay.
Over to you, Pablo.
Thank you so much, Professor Des. I'm busy reading the questions as well while I'm listening. The next question, Dr. Beatus, you have another contribution before I go to the next question.
Yes, no, that is clear, and thank you, Prof, for the explanation on that.
Sure, thank you so much. So this one is from Deriba Abera. He's saying, thank you so much for your nice presentation. For PCA determination, which PF region did you use? Pfcsp or other regions? If Pfcsp region, the overlap of PCA result, was it due to the gene flow or similar response from human immunity? That's from Deriba.
So I can say that we use the PCA based on the Pfcsp gene, and what we are present here is due to the gene flow across the population. And as Professor Ishongoma mentioned, the other papers will come out, which also use different other regions, away from this Pfcsp, but other regions, which at least we observe the same structure, that we didn't find a clear differences, or we didn't see the population structure. So what we have seen here, we also seen the same finding in the other region by use the other part of the genome, which is not a Pfcsp. So what we are seeing is due to the gene flow, the mixing of the gene within the population. Thank you.
Thanks, Beatus. The next question is again from Dr. Richard Pearson, who says, your network slide suggested 3D7 and NF54 have different CSP sequences, different haplotypes. Is that right? If so, this surprises me, as I thought 3D7 was a clone of NF54 of NF54, and therefore I would have expected the 3D7 and NF54 sequences to be identical. That's from Richard Pearson.
Thank you for that contribution. Yes, that is what we were expecting, that the NF54 and 3D7 is the clone of one another to have the same identical. But from what we've seen, that the 3D7 had, when we compare the haplotype diversity and what we got, it has a slight difference between the two sequences. But that is what we got. Thank you.
Okay, thank you so much, Beatus. We still have about 8 minutes to go. If anyone has more questions, please do raise your hand. We will allow you to speak, or type in the Q&A box, and I'll read them for Dr. Beatus. But just to say, Dr. Beatus, the chat box is filled with people saying, really great presentation. Thank you for a detailed presentation. Thanks for the nice presentation. It's just everyone really applauding you, because this was a great presentation and we do appreciate it. But to the audience, as I said, we have about 7 minutes to go. If you have a question, please raise your hand or type in the chat box. Looking quickly. Great talk. Thank you. Thanks a lot. Yeah, it's just really people singing your praises. So I just want you to know that if you can see the chat yourself. Okay, I'm going to give it a minute to see if there are any other questions or comments. I see Saranjeru has raised her hand. Saranjeru, please go ahead and ask your question.
Oh, thank you so much for the presentation. I think the question on resistance, which I would also like maybe an answer, maybe the person was asking that particular gene that codes for this gene, for this protein that is targeted in a vaccine. At any point, does it develop resistance? Or maybe at what point does it develop a mutation such that this particular vaccine is not going to be effective?
Thank you. As maybe I can say that from the background, that the gene, it has the two regions, the N terminal and the C terminal. The C terminal is made up of, it was comprised of some amino acid, like 19 or up to somewhere closer to 100, which this undergoes several variations. And as we know, for the vaccine to work, it needs to stimulate immunoresponses. So if it doesn't stimulate immunoresponse, that it means that you can use the vaccine and it doesn't respond, cause any immunoresponse, or it can cause immunoresponse, which is not specific to what is can help to clear out the malaria. So that variation can take some, because that is very high variable region. Yes, it do happen. And for how long, I can't tell. But the only way you can say that now the vaccine is not working or it doesn't work is when that either it produces a low immunoresponse, or the response which cannot protect the infection. Because the aim here is to protect the infection. So if it does not produce immunoresponses, which is will be enough to clear or to prevent infection, then at that time, no, this we have the vaccine, but it doesn't work. Thank you.
Okay, thank you, Beatus. The next question is from Ainalem Mandefro, who's saying, thank you, sir, for the excellent presentation. The question is it feasible to design a region-specific vaccine based on the circulating haplotypes? What is your suggestion?
I think that if we can come up with a regional, instead of having a global vaccine, which maybe from South Asia to South America to Africa, which have different level of malaria transmission, with some are very high endemicity, some are very closer to elimination stage. So there are those kind of variation. So I thought we need to think coming up with some regional specific. So this regional specific can targeting the available, the circulating haplotype in a given population. That can be, I think that can be ideal vaccine rather than depending on the vaccine which is targeted for the global audience. And that's why we recommend that every country, maybe each region can have this kind of study so that they can have the data at a country level audience instead of the global audience. Because this country, maybe this region have different level of transmission, difference of level of maybe some are closer to elimination, some are still very high endemicity. So that's why we think if we come up with a regional specific, that can be a good vaccine. Thank you.
Thank you so much, Beatus. So there's a question from Professor Dias towards Richard. Richard is answering it in the chat. So that has to do with the PF Haplo Atlas app that has recently been released. But the question towards you is from Saran Jeru, who says, is it possible to design a vaccine that has both CSP and merozoite proteins together to induce a stronger immune response? Thank you.
Yes, it's possible. And there are a lot of technology. As I mentioned before, there is this technology called in silico technology. In silico technology, you can design a vaccine on your computer before you even go to the field. You do the designing, you combine the two vaccine. You can test even the immunoresponse based on the computational. So that's why it can help you maybe to come up with this vaccine when you compare maybe AMA1 or other vaccines, CSP, how much you're going to respond. So when you go for the real work, you can already finish most of the work. And this is maybe the very good development. Instead of designing a vaccine and starting from scratch, you can finish maybe 50% to 60% on your computer. There are a lot of tools which are available, which help to design drugs, to design vaccines, to design everything that you use your computation approach. And from there, you can go to the field or to the lab and do the testing. So yes, it's possible to do that. And you can test it or you can do it in silico before you go to real work. And instead of using a lot of money doing the lab work, you can use maybe small amount of fund to do the computational part and see the feasibility. You see how much it performs under computer before you do the real work. Yeah, that is my comment. Thank you.
Thank you. I'm going to try and squeeze just one more question. Apologies, we are approaching time. We have a minute left, so I'm going to squeeze this one in. It's from Abdallah Juma, who says, great presentation. How long is the shelf life expected for RTS vaccine to induce a sterile immune response in human beings? That's from Abdallah.
So I think there are several studies coming out from the several clinical trials, which have indicated how long exactly the vaccine can take to induce the immune response. I can't tell exactly the days or the time, but it is there. It can be found. We can get this through the different clinical trials which have been working on the RTS vaccine. Thank you.
Hey, thank you so much, Dr. Beatus. I think, as I said, Mohammed Sani just said, thanks for your comprehensive presentation, and I concur. Really, this was one of my most enjoyable journal clubs. that we've
Thank you.
hosted. So thank you so much and really well done. I can tell you did the work and you prepared and you really gave a well-explained and accessible presentation. So I just want to say thank you so much for your time.
Thank you.
We are really grateful and we'll keep in touch. And for those of you that have more questions, please do email Dr. Beatus, follow him on Twitter and on LinkedIn as well, and then you can get in touch. Dr. Des, someone can share the link on here. So Dr. Des is asking about the Haplo Atlas app that has just been released. Someone will share the link with you, Professor. And to all of you, I just want to say thank you so much for joining this journal club for July. In August, we're going to have 2 journal clubs. We're going to have one on the 6th of August, which is going to be presenting on the new app that Professor Des Ishingoma is asking about, which is a Haplo Atlas app that has been released recently. And you hear more about how to use it and what it's for. If you check the Q&A, Dr. Richard Pearson and Christina have been also talking about it as well. So you'll hear more about it from Trin and Nina on the 6th of August. And on the 27th of August, we are going to have a journal club from Dr. Kelly Bennett. So looking forward to those. Please do check out our social media as well as our website for the times and the links to register. And again, Dr. Beatus Lyimo, this was wonderful. Thank you so much. And everyone, stay well and see you next month. Thank you so much.
Thank you very much. Thank you, Maria Jenny, again for giving me a chance to present. That was another good experience to share what you have findings, the global audience. And thank you much for the comment. We look forward to keep up the good work. Thank you and bye-bye.